Hello and welcome to another episode of An Unexpected Fight. We are so excited today to have Dr. Gojo here with us to discuss some rare brain tumors. Kelly, you want to take it away?
Yes. So today we have Professor Johannes Gojo, who is a leading physician scientist in pediatric neuro-oncology at the Medical University of Vienna. With training in medicine, neuroscience, postdoctoral work at Dana-Farber Cancer Institute and the German Cancer Research Center, he has dedicated his career to advancing precision medicine for children with brain tumors. His research focuses on high-risk pediatric brain cancers, including ETMR, ependymoma, and high-grade gliomas, where he develops innovative tumor models and liquid biopsy techniques to improve diagnosis, monitoring, and treatment. Professor Gojo leads international collaborations such as Care for Rare, which was established by Solving Kids Cancer, and brings together the ETMR/ATRT consortia. He is widely published in Cancer Cell, Nature, and Science, and is deeply committed to ensuring families' voices are included in research. With compassion and innovation at the heart of his work, Professor Gojo is helping push the boundaries of what's possible in treating the rarest and most devastating childhood brain cancers. I think it's pretty obvious why this is close, near, and dear to Tiffany and I's heart, so we're very excited to have you today.
Thank you very much for this kind invitation. The excitement is fully on my side as well. It's a pleasure that I can join your podcast. Thank you.
So, Dr. Gojo, your early work in dissected tumor heterogeneity used single-cell sequencing. So why is this such an important breakthrough for understanding pediatric brain tumors?
So thank you very much. So we were really excited when this technology came available because as you all know, tumors are diagnosed by their histopathological patterns, which inherently also already tells us that there are different cell types within a tumor, so that this tissue is distinct, that there's a distinct architecture within these tumors. And this technology gave us the possibility to really look at the transcriptome of those single cells and see how they orchestrate the malignant tumor tissue. So, and transcriptomics obviously is one part of messaging within a cell, and this gave us a lot of information. Meanwhile, there are a lot of more technologies available, of course, but it still is an integrative part of our work.
It's definitely fascinating. So you've trained all over the place. Looking back at your training in Vienna, Boston, Heidelberg, what scientific approaches from those labs most shaped your current research direction?
So I think overarching all of those fantastic centers is their compassion for translational research. So of thinking from the bench to the bedside. So those are all centers that are close to patients, close to development of therapies, but also close to innovative technologies such as single cell sequencing. So which I learned in Boston, of course, where always novel technologies are driven early on, but always with the idea of how can we translate this into novel treatments? What does it tell us? How to stratify patients, how to pick the right patients for the right therapy.
Love what you guys are doing. So cool. We've gotten to talk a lot, obviously, at the Care for Rare consortia that we went to in February. And that consortia discussed how to better treat ETMR and ATRT. So can you kind of tell everybody like how that was structured scientifically and why a collaborative model like that is essential for ultra rare tumor research?
So the unique, but I think more and more modern way this consortium was shaped, that it was driven by patient representatives. So it was really patient representatives or organizations like yours that brought together key experts in the field. So a very open discussion, and what I think was really also central to such a meeting and the other meetings that were held is that it's open in between the experts. So that these are rare tumor types, and it's very important to drive innovation together, to share information early on, to really build upon the blocks everyone is bringing in this research. But it needs a framework. So because of course it's complex, it's complex science, it's complex clinical studies that are being developed, and therefore such a framework, bringing people together, discussing the insights into those tumors from early research to biomarkers until how do we think realistically can we develop a clinical trial is very, very important. And also further on, there have been novel kind of approaches to connect, etc. So it was really fostering a network that is ongoing and aiming to develop, on the one hand, consortia for basic research, but also clinical trials, which is a real challenge in those rare tumors, especially if you think of global studies.
What I found fascinating while we were there is that, I'm from the US, from Austria, I had people there from Italy and just all over the world, and to hear how differently you all treated ETMR children and how that translated into outcomes, really, because in the United States, ETMR, the outcomes are horrible. But it sounds like in other parts of the world that they may not be as bad as they are here. And so it's really cool to get everybody together and kind of say, hey, let's make this collaborative approach. That way we're working on this at a more unified level that's going to work better for the patient. So that's one of the things that I found that was really cool about hearing everybody sit around and talk.
Yeah, thank you. I mean, importantly, we have to acknowledge that it is a very aggressive tumor type, and that's also particularly interesting about these collaborative approaches, and also about these communities, that everyone acknowledges that they do not have the one answer on their own, so that it needs collaboration, it needs different ideas and concepts that then are being brought together.
Absolutely. Tiffany and I have talked to many, from different consortias about data sharing and the importance of it. Can we dive into that? Like, what kind of data, genomic, transcriptomic, liquid biopsies, like what's being shared, and how does that accelerate discoveries compared to working alone?
So whenever possible, data is shared early on to allow other researchers to work on it. But this, of course, has also legal frameworks that we have to adhere to, also in an international context, of course. And this can be any type of data. But the general the general way how this is dealt with today, also in the whole scientific community, is this: once the data is published, so also checked by peer review, it becomes available publicly in a data protective way, of course, because you always have to respect individual rights as well, with respect to data protection, so that everyone can benefit from it. And we have really built upon case series and data in a stepwise approach, also in the past, in these and other rare tumors.
Awesome.
And I think importantly, again, in this collaborative way that we have also fostered in the Mexico meeting, you connect the people and enable such exchange.
Yeah, I think that was an awesome thing, because you really don't know who's working on what until somebody says, oh, this person's working a lot on that, this person's working a lot on that, and then you can make those connections to be able to share that data or call each other and say, hey, got this difficult case, what do you think about this, what are y'all doing here? And it makes it a really great way to foster collaboration and to get better treatments for all these kids. So, go ahead, Kelly.
I think that's what one of our goals is with the podcast, even just,
Yeah.
getting the word out there. People just really don't realize, who's out there, who's doing what. And we're getting better as a country, I think, in general. But until, recent years, I think the collaboration was less than what it needs to be, and it still can improve. But I think, that's why Tiffany and I are so passionate about just sharing all the research we can and connecting people. And hopefully somebody's listening to this saying, you know what, I want to support Dr. Gojo now.
And I mean, also by supporting a network, and to support actually also many other efforts, because that's the main thing about the network. So that's the excellent thing, still.
Absolutely.
So you have a newly funded research grant that has been partially supported by my foundation, Gold Ribbon Kids, and I feel like this is a major step forward for ETMR. Can you walk us kind of through your ARMOR trial, what your hypothesis is, and the scientific question that this project is trying to answer?
Yeah, thank you very much. So first of all, thank you very much for all your efforts of making such funding possible. So it's I think those grants are essential to drive also innovation and to foster other projects, as I said, in the whole international network. So we're very excited that this project was selected, and the aim is to build upon what we have already learned about architecture of tissue of ETMR, about different cell types that are in there, and to bring this at first in the context of the architecture of the tissue. So we use the next development, let's say, after single cell sequencing, which is spatial transcriptomics. So we can really see where the cells with certain properties sit in the tumor, and this allows us better to see how those cells interact and which mechanisms really shape the tumor as a whole, all with the idea to identify interactions that can be used to treat the tumor and to attack those cells. So the idea is, and then in the next level, we want to compare the primary tumors to the recurrent tumors, because these we assume are the ones that are selected for the bad guys. So to in a simplistic way, and to see which are those, what are their properties, and how can we better attack those, and maybe also how do they look like in the first place, so that we can attack them efficiently in the first place, so at upfront therapy.
And how are you using, like, what experimental models or technologies will you be using for this? Like animal models? How are you getting to this?
So we will be first focusing on patient tumor tissue that we have available at our center, but also within international collaborations, of course. So this in rare tumors is very important, of course. And then at next we have developed in vitro models that are better for screening. But here also it's important to emphasize that those are very rare for ETMR, and that within such consortia everyone is working on developing those, and then hopefully also sharing those. So we aim to develop novel models out of patient tumor tissue and make use of the ones that are already established. And then on the next level, as I have indicated, we are looking at the whole architecture of the tumor, which also includes brain tissue. So it's a tumor that is in the brain that interacts most likely with brain tissue. So then we have already done this and will propagate this in this project to fuse tumor cells with brain organoids to really mimic in the model the architecture of the tumor, and to see whether those hypotheses that we generate from the patient tissue for therapies hold true in such a multicellular setting. So to really at best model this, and then ultimately the best ones we would also aim to check in mouse models, which is kind of the gold standards for checking therapies.
I find it fascinating, especially in ETMR, listening to everyone talk so for those of you who don't know, they can take tumor cells and basically replicate them and grow full cell lines to where they have extra tissue from, granted, it's a single patient per cell line, but you go in and they're able to do testing on actual tumor tissue, which is really cool. It's great technology, but the hard thing, like you said, with ETMRs, there's just not a lot. I think that in the consortia, they said that there was 7 cell lines or something like that. So all of these researchers are trying to work with basically 7 ETMR patients, so to speak. And so that makes it kind of tough. I mean, I was going to donate Nora's tissue for research, and because she had been diagnosed with COVID 9 days prior, but was asymptomatic, every pathologist and every lab in the state wouldn't touch her. And so that was wasted, in my opinion, a wasted opportunity for growth and for furthering science. But, you know, it is what it is in that regard. However, if you're out there listening and there's any tumor tissue that you have, like if your child has been diagnosed with one of these ultra rare tumors, like you can do things like donate that tumor tissue for science to be able to further what these researchers are trying to do to try to better these therapies that our kids are getting.
I understand, and we're, of course, and of course the whole network is always very, very grateful for these donations, and luckily there also meanwhile exists assistance, I think, across different countries, if there's the wish to donate tissue to help with it, because it again has to also adhere to the national legal framework that exists in the respective country. So it's good that there is guidance meanwhile.
So ETMR and ATRT are probably some of the most aggressive brain tumors. So from a molecular standpoint, what do you think makes them so resistant to therapy?
So what we have learned about those 2 tumor types, that they have very distinct drivers, which also kind of marks them as very specific tumor types and unique tumor types. And unfortunately, these tumor types tend to recur and evade the therapies that we can offer today, that are the standard therapies that are curative in other brain tumors, so that they may work in medulloblastomas, but they may not work in a proportion of ETMR and ATRTs, but also do not work in a proportion of medulloblastomas, for example. So, and I think that's a central goal of research around the world of many, many excellent people, to identify those factors that actually enable those cells to evade those tumors. Because frequently, in the first place, they also respond, those cells die, but some cells eventually survive this first therapies and then come back, and to identify those mechanisms that characterize and enable those cells to kind of survive these therapies. This is essential to come up with next therapies and ideas of subsequent therapies, of course.
Absolutely. So you've kind of touched on this, but just to maybe elaborate a little bit, how is your team working to identify these vulnerabilities in these cancers, so we know that these cells are very resistant to many standard chemotherapies? But is there anything else you're working on, whether it's like DNA repair pathways, epigenetic regulation, metabolic targeting? Can you touch on some of that?
So in the first place, many other labs as well, they compare the primary to the recurrent tissue and check whether there are any obvious differences that maybe point towards a certain therapy. And then when it comes to distinct pathways, for example, for DNA damage, we do combine certain compounds that have maybe also been developed for other cancer types with irradiation or with chemotherapy, in order to see whether this makes it more efficient, at least in the lab. And this can also be true for other therapeutic concepts, for example, for targeting certain pathways with small molecules that inhibit the signaling. But importantly, what we aim to do is to combine treatments, because we are convinced that a single treatment may not be efficient and is more easy for a cell to evade and to adapt to, and that combination treatments may be more efficient and especially more long-lasting in their effect. Absolutely. Importantly, in the next step, of course, this has then to be taken into translational research and checked whether this is tolerable, this combination. So, of course, it has to be clinically tolerable.
I'm going to throw...
Oh, go ahead.
I'm going to throw you off with this one because it was kind of a little off topic, but it's just something I was thinking of. So we're obviously, collecting samples of brain tumor, kind of studying, looking closely. Has anyone thought to try to find patients, like my child was an outlier. Obviously, she's a stage 4. They called her glioblastoma back in 2020, but now it would be considered an infantile high-grade glioma. But she's 5 years out, doing fabulous, has no evidence of disease. I often want them to look deeper into her particular sample of tumor and see why. Why is she an outlier? What put her? She had no genetic markers that were significant, a couple variants of uncertain significance right now. But have they ever thought to collect samples maybe from these kiddos who are doing well and are surviving and compare them to the children who did not and see, if there are any outliers that we just by comparing those cells side by side? Is that ongoing at all, research?
That has been extensively done across the whole group of pediatric brain tumors in the past decade or decades already, I must say, with molecular reclassification. And one outcome was, of course, to identify ATRTs and ETMRs as distinct tumor types, unfortunately with dismal outcome, but we have also identified other tumor types that were lumped together previously with others that have better outcomes. Yeah, for example, in medulloblastoma, we have identified different molecular subgroups, some of which have a better outcome than others. So this is ongoing work, especially with molecular classification, as you mentioned, to check on the tissue and to see whether there are differences. But this requires very stringent data checks, let's say, that you really can compare it in a confident way, and also with confidentiality, because there's personal data. So this is why it's also important for those other tumor types like ATRT, ETMR to come up with clinical studies that have then a structured availability of tissue, structured availability of homogeneous treatments, so that you can really identify those different types that may be there. I hope this makes sense.
Yeah, no, absolutely. Absolutely. Thank you.
So do you feel like most of the research that's ongoing for pediatric brain tumors are utilizing still the same old types of chemotherapy that have been around for decades, or are we looking at new therapeutics, new targeted agents? Are they considering any of the things that are being approved for adult cancers, especially in like more targeted themes like an EGFR, an ALK, an NTRK? If one of these brain tumors were to have one of these mutations, would those be considerations for therapy?
So I think we're doing both, let's say, so with more molecular information or more also other certification methods for pediatric brain tumors becoming available. We are also rethinking our standard therapies to be more precise. So I think it's also always important to acknowledge that this is also part of precision oncology. So not only thinking about the novel agent or a novel target, but also how to rethink therapy for the standard therapies and to do new stratification methods or new stratification biomarkers. And when it comes to novel agents, we do have novel agents available that have become available in the past 10 years, as you mentioned, targeted compounds like NTRK, ALK inhibitors that are being used if there is evidence that there is a target that can be used. So not to use a drug when it's considered to be useless. But this is, of course, a small proportion always, but it's important that this is now considered all about in few patients, but to be effective, and that they have come available. And I think this is a real success. Still, what we aim to do in the pediatric neuro-oncology world, I would say, or consortia, to not stop thinking about it. So to be kind of satisfied with it, there's a novel agent, we can use it, so that's it. But really always to think ahead of how will we integrate this into a full therapeutic concept with the other therapeutic strategies that we have available.
Yeah, I know DFMOs.
Out of all the Go ahead, Div.
DFMO's been pretty widely adopted now for ETMR. Is that utilized in multiple brain tumors? Is that really just in ETMR?
DMFO, if I just should.
Yeah.
So no, I'm not aware whether it's also used in other pediatric brain tumors, but of course in ETMR it is widely used. Not yet, as of my knowledge, in a structured clinical trial. So it's still difficult to assess what is the real effect of DFMO.
You talked a lot about the different biomarkers. Are there any that look most promising right now? And how close are those to clinical application?
If we think about the whole spectrum of pediatric brain tumors, for example, in infantile hemispheric gliomas, the fusions of NTRK or ALK have been identified in the subsets, and I think the important task is to investigate their value or their position in the complete therapeutic concepts, and importantly in many recurrent or also upfront cases, they have shown that they have value, and fortunately there are now trials ongoing comparing it to our previous standard of care. So these are definitely biomarkers that have worked when kind of matching a target to a therapy, let's say. And then there are, of course, other biomarkers, for example, like the medulloblastoma subgroups that have already entered the stratification for therapies. For ETMR, I think the most important step was to identify the tumor at all in the first place. So this was still very challenging a couple of years ago to really identify this tumor. And then although we have limited knowledge about the optimal therapy, to really then at least go ahead with the limited knowledge that we have for therapy, and not to mistake it for another tumor type and give something completely other therapy. I hope that makes sense. But it's of course the aim of such consortial efforts to identify biomarkers that may even help in stratification within those rare tumors.
So you studied the role of telomere maintenance and oncogenic drivers in these tumors. So what do you see as kind of the biggest opportunities for targeted therapy in the future?
So we have, for example, studied telomere maintenance in a certain type of high-grade gliomas that have certain alterations that enable those cells to elongate the telomeres, so the end of the DNA strand, in order to survive multiple cell division. So this is a very central characteristic of tumor cells. And interestingly, we found that this confers hypersensitivity to DNA damage inhibition. So this mechanism of survival of the cancer cell is accompanied by sensitivity to certain DNA damaging compounds. And I think identifying such connections opens new therapeutic options, and we can rethink therapies for certain tumor types if we identify this. All of course, still in the lab, but then potentially in translational research.
So talking about go ahead.
Targeting other telomere maintenance mechanisms, for example, the TERT promoter mutations that are present in some high-grade gliomas, are more difficult and challenging.
So we know it's different, obviously, finding out these things in the lab and, really getting to the bottom of it versus applying them in a clinical trial. So how do you balance the preclinical discoveries with the need to translate them into an early phase clinical trial?
So luckily, there are also precision oncology efforts now ongoing in various countries that enable upfront, or not upfront, but early administration of innovative therapies if it's deemed justified, let's say. So it's always important in the clinical context to think about the whole clinical case, about comorbidities, potential toxicities, age of the child, and also, of course, the overall wish of the family and the patient on how to proceed and how they want to shape therapy and the life around it. Let's say I think that's always important to consider to come to an individualized therapeutic approach based on the evidence there is. And luckily, there are now ongoing efforts like drug screening studies or precision oncology studies that add additional layers that can be used for therapeutic information. And we use all this then, of course, to shape clinical trials that then give us a much better evidence of those therapies. But this, again, cannot be done by single centers, but all within networks, because it's extremely complex. There are big regulatory demands to proceed with such trials. So I think that's why it's very important that those networks are also supported, because only they can really leverage this task.
So what are the biggest challenges in designing a clinical trial or research, such as the ARMOR trial for ultra rare tumors like ETMR? Is it small patient numbers? Obviously, we know that's an issue with these ultra rare tumors, but endpoints, regulatory hurdles, is there anything else that makes it difficult to do some of these studies?
So within our ARMOR project, we of course aim to generate solid preclinical evidence for potential clinical trial, and we aim to provide this on multiple levels, so that there's like I mentioned initially, with mouse models, with other in vitro models, that we really have a good idea and are confident that the potential therapy would work in patients. And then the first main challenge, of course, in rare tumors is patient number to reach really reliable data. But luckily, different consortia provide guidance also with statistics and innovative statistic concepts to really be able to do trials with smaller patient numbers, which is still challenging. Then it's the second hurdle, of course, is conducting trials in an international context with different legal framework that needs a lot of help and guidance. What I do not think is limiting is the motivation of families, patients, and experts. So those networks work really, really well, and are really kind of trying to move things forward. What's of course challenging within those networks is to come to consensus which steps you would follow next, because of course we would like to answer all questions at once, but this may be not possible in rare tumor types. So you have to come to consensus of how to stepwise approach it.
And not to throw you on the spot, but can you give us an example maybe of where like a lab discovery directly changed how you treat a patient or design a trial?
So one example would be, for example, targeting PDGFRA in high-grade gliomas. So different labs discovered that this was a receptor tyrosine kinase that's active in high-grade glioma, and there were different drugs used, not that efficiently, but recently we could also publish a study together with the lab of Marielle Philbind and the lab of Karl Koschmann in the United States, that another drug that was actually initially used for other type of disease could more efficiently target this receptor and was also going into the brain. We do acknowledge that a monotherapy with this is maybe not kind of curative or not kind of the full story, but at least this came to the development, even that then based on this data, a clinical trial was already developed. So this was actually, until coming to the clinical trial, it was really a success story of also how fast ideas can come from the lab to the clinical trial. Of course, within clinics is also always more difficult, and we have to find again where the value is of this new asset, let's say, in therapy, and this can be a longer process than just discovering it in the lab, but at least we're one step further.
Thank you.
So, kind of going back to the international networks like Care for Rare, this is invaluable for collecting data sets. So, do you see machine learning or AI being used to analyze some of these massive data sets that we can collect?
So I'm obviously not a data and AI expert, I have to admit, but I do see that this is more and more used and will become and has become already of much value also to boost innovation, because it may enable us as other novel tools to find new connections, to generate new hypotheses, and also to select the best hypothesis we then validate in the lab, for example. So for example, to identify new drugs or drug combinations that are reasonable. But as you mentioned, it only works if the data is available. Mm-hmm. And so this is again points towards collaborative efforts, of course.
Yep. And for any families out there listening, this is why it's very important to advocate for genetic and genomic testing and sequencing, because if we don't have those results, that's just less data that we have that eventually, I personally, again, I'm also not an expert in AI, but I do feel like if we can get enough data out there, eventually we'll start to see some overlap, and we'll start to see some patterns that hopefully the very smart researchers out there of the world will be able to break down and analyze better.
Now, this is kind of off topic. Do you feel like there is a good utilization of genomic and hereditary cancer testing in the pediatric world? Because I feel like in the US, the utilization of this technology is not there. I had to beg for my daughters to be genomic tested, and that shouldn't have been, but obviously we're also dealing with things like really small tumor samples from some of these hard to reach brain masses that these children have, because you just can't go in poking all over your brain. So I understand that there's probably limited tissue to be able to do some of these testing, but do you feel like in other parts of the world, especially where you're at, do you feel like you utilize genomic testing maybe more than we do in the US? I think that what I hear is they do relapsed and refractory profiling, but as far as initial sequencing, it's not done standard on the front end.
So I think there are excellent centers all around the world. But the question so maybe for the first part, for hereditary cancer, this of course has additional ethical and legal implications as compared to testing of tumor, which are of course then also individual decision. So that's always important, and this is why it's maybe also differently handled as identifying a tumor type based on analysis of tumor tissue. But it's of course increasingly being used and aimed to additionally inform management of patients. This is particularly important, for example, in ATRT, where a certain proportion of tumors arise based on hereditary predisposition, and this changes the management and follow-up of patients. So, but still, I think this is a broader discussion whether more wide use of genomic testing is justified or not, because it has, of course, many additional implications.
Right. We actually did a whole nother podcast episode on that.
Yeah, I assume it.
It was a whole episode. So if you want to hear more on that one, go take a listen to that one. All right, Dr. Gojo. So if we look forward like 10 years ahead, what's your vision of how pediatric brain tumor care might be transformed? Will it be more personalized, hopefully less toxic? But just in general, how do you believe it'll be fundamentally different?
So I hope, let's say, to put both ideas into one, yeah, I hope we will better know for who more toxic therapy is justified and who benefits from it and who doesn't, like we already do know. Yeah, so we have already advanced here. And if we apply toxic therapies, I do hope that we will be able to shape them in a way that they are more efficient or more directed to the correct therapy. So I fear that we will not be able to reduce toxicity completely, but I do hope that we will maybe be able to manage some toxicities better, and also to adapt the therapy to the individuals. And then of course, I think that this also comes together with more personalized treatment, but this has, of course, to be balanced against our knowledge or our confidence into treatments based on comparison to other cases.
Absolutely.
So finally, for parents listening who may be deep in the fight, their kid has a brain tumor, they're going through all these therapies right now, what message would you want them to take away about the direction and hope for current research?
So what I always tell patients, or what I fully do understand, is that this is at first, the first diagnosis, it is a shocking experience, and it's mostly a desperate situation, and that they seek any help they can get. And frequently, I learned that this can also lead or enhance confusion that is anyway there, and that's why I think it's very important that patient and parent organizations are there to guide. And also I encourage that there is confidence based on networks that doctors do connect. Yeah, so if patients are connected with the right center, those centers also connect to other centers, particularly in those rare tumors, to really inform and come to the best decision for treatment based on the whole network that is there. And this is what we experience. Here, of course, technology has helped us a lot. So such as we are now discussing in a virtual podcast, we are also discussing cases virtually now across the whole globe to really get, fortunately, the latest insights from the whole network in order to best inform treatments in rare cancer. So there I also encourage confidence, that the doctors and physicians and our whole treating team is also taking care of getting this information, at least from the centers that we are connected with, because I frequently experience that then parents have the feeling that they have to get those pieces of information from every single center. And then actually I experienced that they have already connected on a professional level.
Absolutely. And hopefully the fact that, I mean, we're communicating with you all the way from Austria and on this podcast, it gives hope just in itself to show that, you know, yes, it's a big world that we live in, but, you know, some of the brightest minds are coming together and sharing their research, even with two cancer moms like us. So we appreciate having you on here so much and for your time. We know how busy you must be.
Thank you. Yeah, I think it's really all bringing things together. So I think initiatives like yours, like information, like your podcast, are really, really important pieces of a whole puzzle that brings together different aspects and important aspects in advancing the field, but also in supporting families and patients.
Absolutely.
So much for on today. So excited to see the armor trial or preclinical work to get started and see where that takes you. I think that you're an amazingly bright mind, and I think that this research is going to go far, and I'm really excited about that. If you guys want to get in contact with Dr. Gojo, we'll put his contact info in the show notes below. If you want to talk to Kelly or I, our emails will also be noted there. If you want to look more into his research, we'll link a website below. For me, you can reach me at goldribbon-kids.org or Kelly at nationalpcs.org. And as always, we look forward to seeing you again.